Okay, so today we are going to go over the phases of mitosis, what happens in them, and everything that changes in the cell as we go through all those different phases. So last time, guys, we went over just what is going on, what is our goal of everything, and that's going to be to make a new cell, to replicate a cell. In prokaryotic cells, the goal would be to make literally a brand new organism. In eukaryotic cells, maybe we're renewing something, we're replacing something, or something happened where you got hurt or something like that, so we need to fill a hole. So today we're going to go over, like I said, all the steps. Now, last time we talked about the number of chromosomes that we have in our somatic cells, and that was 46. Gametes are going to be half that number. We're going to have 23 in there. So we want to make sure during mitosis, during our cell division, that not only do we replicate the cell, but we also need to replicate the nucleus and all the contents inside the nucleus, all those chromosomes, because we don't want to replicate a somatic cell that originally had 46 chromosomes and then half that number, so we're only going to have 23 afterwards, because that's no longer a somatic cell, that's a gamete instead. Okay, so along with copying and making a new cell, we're going to have to copy the DNA as well. So we're going to divide our cell cycle for the reproduction of somatic cells into two different parts. We're going to have the mitotic phase, or mitosis, and then we're going to have interphase. Interphase is when we are going to take that DNA, when we're going to make a copy of that DNA, and also we want to make sure, along with making a copy of it, we want to make sure it's a good copy. We do not want a bad copy for that where there's mistakes, because that's, we'll talk about it later, but that's bad. So we're going to start off with interphase. Most of the time, your cells are in interphase. Now, it's different for different cells in your body. Brain cells, especially when you're my age, they're not going to be doing a whole lot of reproducing anymore. Okay, mine are kind of just, they're just vibing up there, you know? So other cells in your body, maybe ones that are working harder and maybe working more often, they're going to be in interphase a little less of the time, and they are going to be in mitosis more often. Maybe skin cells. Skin cells reproduce often because you lose, your outer layer of skin like, every 30 days or something like that. So depending on the cell, some cells are in interphase more often, some cells are in interphase less often. Interphase is when the cell is not reproducing, okay? So we're going to have three different parts. We're going to have the G1 phase, it's called gap 1. We're going to have the S phase, that's going to be synthesis. And we're going to have the G2 phase, which is the second gap. During this time, the cell is going to be getting ready to reproduce. It's going to be getting ready to go into mitosis. So the cell is going to be growing, and along with this is when our DNA replication is going to happen. The DNA replication happens in S phase, okay? And that's what we mean by synthesis. Synthesis, we're talking about synthesizing the DNA. So here is interphase. So this is, we're going to go over just the steps of interphase. You could see that most of this is your cell cycle, most of it is interphase, and then this small part here is mitosis, okay? That's when the cell is reproducing. So here's what happens with everything, guys. G1, the cell just got out of mitosis. We have this brand new cell. So the goal of G1 is for the cell simply to grow. It wants to get bigger. It wants to get to be the normal size of a cell. Now, if the cell is going to go into mitosis, if the cell is going to reproduce, it's going to carry on over into S phase. If the cell is not going to reproduce, like the brain cells I was talking about a little bit ago, it's going to stop right here, okay? And it's going to chill in G1. Okay, so it grew, it got to be the size of a normal cell, now it's a mature cell. If it is not going to reproduce for a while, it's just going to chill in G1, okay? The other place it could go, which is not in this diagram, which I wanted to share with you guys, right after G1. So G1 is just a cell growing up. It's becoming an adult. Right here, in between G1 and S phase, there's also a G0 phase. Okay? And this is a place where the cell goes to literally stop reproducing. All right? The way I always describe it, it's like the nursing home for cells. But in a nursing home, I mean, when you guys think of a nursing home, you think of like older people just kind of chilling there. They're not doing too much. In G0, yeah, they're just chilling there. But the only thing they're not doing is reproducing. They're doing everything that a normal cell would do. They're functioning properly. They're going through every daily activity perfectly fine. They're just, I'm done reproducing. I don't need to do that anymore. Okay? So that is, if they enter G0, that cell is done reproducing. It doesn't reproduce anymore. All right? So let's go back to our interphase things here. So G1, our cell grows. If it is going to reproduce, it's going to carry over into S phase. And this is where we replicate our DNA. Okay? So what we're going to do here is, if we're talking about our somatic cells, originally, if you remember, we have 46 chromosomes. If we replicate it, we have a copy, so that's the second copy of it. So now we're going to have 92 chromosomes altogether at the end of S phase. Okay? So 92 at the end of S, once DNA replication is complete. Right? After that, we are then going to move to G2. What's our purpose of G2? Well, we want to make sure, guys, and as I talked about this in the previous slide, we want to make sure that our DNA is perfect. We do not want any mistakes in our DNA. So what we're going to do in G1 is we are going to, once again, continue to grow, just like we did in G1. In G2, we're going to continue to grow, and we're also going to check the DNA. Check DNA for mistakes. Okay? Why are we checking the DNA for mistakes? Because if there are mistakes, that can lead to a C word that we really don't want, and that is cancer. Okay? Cancer could be a mutation in our DNA, and that could lead to our cells just reproducing. And that's just one mistake, guys. Every time your DNA makes a mistake, that does not lead to cancer, but that's one thing that it could lead to. Mistakes aren't always bad. Mutations aren't always bad. Sometimes with mutations, they lead to good things. Maybe it's some kind of phenotypic or something you guys can change in your body that gives you an advantage with your DNA. Okay? So mutations aren't always bad, but sometimes they are. Some mutations actually even lead to evolution. So here, if we get into mitosis, we went over interphase already. So that's when the cell is not reproducing. Now we're going to get into mitosis. This is when the cell is reproducing. And then later on we'll get into cytokinesis, and that is kind of the cell just dividing things up so that it has all the parts it needs. Okay, so we are, in the first picture here, we're in G2 of interphase. So the cell is not yet reproducing. When the cell enters mitosis, the first thing it's going to do is get into a stage called prophase. And we'll go through everything that happens here, each one of these steps later on. It's then going to go to prometaphase. I don't believe we went over this in regular biology, prometaphase. We went right from prophase to metaphase. And honestly, not much happens in prometaphase. Very quick step. Next, we go to metaphase, anaphase, and lastly, telophase, followed by cytokinesis, which is the division of everything inside the cell. All right. And these are just the other pictures, guys, that are not under the fluorescent dyed microscope pictures there. Okay. So I'm going to go through all the steps with you guys, but before we do that, if you remember last few chapters ago, we went over the cytoskeleton, and we talked about the cytoskeleton with the intermediate filaments, the microtubules, and the microfilaments. All right, and we talked about like all the dynein walking across the microtubules and all this stuff. We're not going to worry about that stuff now, but we are going to concentrate on the microtubules right now. And in animal cells, we have these centrioles that are in the middle of the cell, kind of right outside the nucleus. Now we're going to use that centriole. And what we're going to do is we're going to form something called the mitotic spindle. And that's going to help to divide our DNA between our two new cells. Okay? I'm going to show you the picture of it here in a second. Aster, it'll be easier for me to show you the picture of it. So here, whoops. Now we'll go with this one. So here, guys, okay, we have our chromosomes lined up in the middle of the cell. And we'll talk about what stage this is later on. But we had our centrioles in the middle, okay? They are going to divide and go to different parts of the cell. They're going to go to the outside of the cell, okay? And what they're going to do is they are going to, it's almost like Spider-Man, they're going to release these microtubules. Come on, lights. There we go. They're going to release these microtubules, and the microtubules are going to extend to the middle of the cell. When they extend to the middle of the cell, they are going to latch on to the chromosomes, all right? When they latch on, the goal is for them to later on act like almost like a fishing hook, I guess you could say. And what they're going to do is they're going to latch on to the chromosomes, and they are going to reel them in. All right, so they're going to separate them. And that's our goal, is to get the chromosomes to separate sides. All right, so now that we know that, let me go back to our pictures. I'm going to go back to these normal ones here. Instead of using the ones with the microscope ones where everything was fluorescently dyed. So during interphase, we replicate our DNA. So let me go through everything that happens in all the steps here. We are going to have our DNA in the nucleus during interphase. Now, our DNA at this time, if you remember the vocab word from a previous chapter, it is chromatin. So this right here, this is DNA in the form of chromatin. Do not get that confused with chromatid, which is what we're talking about in this chapter. So chromatin is a very uncondensed form of our DNA. After this, when we get to prophase, our DNA is going to condense. So three things that happen during prophase. Our DNA is going to condense into our chromosomes. So you guys can see it's not like the spaghetti pasta stuff over here. Now it's forming, I guess, bigger noodles, we could say. But the chromatin becomes chromosomes, or if they're together, chromatids. What also happens is our centrioles that were once right by the nucleus are going to separate into opposite poles of the cell. So one's going to go to the one side, start making its way over. The other one's going to go to the other side. The last thing that happens is our nucleus begins to disappear. So three things in prophase. The DNA condenses to become chromosomes, the nucleus begins to disappear, and the centrioles begin moving to opposite sides of the cell. Okay. After this, we are going to have prometaphase. So during prometaphase, the nucleus has now disappeared almost completely. We are also going to have our DNA completely form into chromosomes. Now, I'm going to try to fit this picture in here. I know last video we went over the chromosome you get from the sperm and the chromosome you get from the egg. And in the middle here, we had our centromere. Okay. So I'm going to erase all that. All right. Along with having a centromere on each one of these, kind of where the X is, like where the X kind of folds there, right here and right here. This structure is called a kinetochore. Right? There's one on each chromosome. So if we talked about chromosome number 1 and it's 2 pairs, we're going to have 2 kinetochores there. Okay? And don't forget, altogether we have 92 chromosomes right now in our cells. So if we have 92 chromosomes right now, it's already been duplicated from our 46. So if we have 96 chromosomes, we're going to have 96 kinetochores. All right. So during prometaphase, guys, nucleus disappears. The microtubules that are going to be released from our centrioles, they are going to connect to the kinetochores. All right. Then we're going to move on to metaphase. Those microtubules are going to line our chromosomes up right in the middle of the cell. Okay, so I always think of metaphase stands for middle. So they line up right in the middle of the cell. And that's everything that happens during metaphase. So we have during metaphase, we have our chromosomes lined up in the middle. Our microtubules from our centrioles are attached to them. And then also, if you notice on the other side, the microtubules actually attach to the cell membrane. And what that does is it helps to anchor the centrioles so that instead of, because we're going to disconnect the chromosomes during the next step, during anaphase, instead of the, let me draw it here, instead of the centrioles coming inward, we don't want these to go in. We want our centrioles to stay put. So that's why they anchor themselves to the cell membrane right there. And then what will happen is they will reel the chromosomes in that way. And the microtubules will shorten a little bit. Okay, so that's metaphase. Next step, anaphase. You can see there is at the kinetochore, we are going to have our microtubules start to reel back toward the centrioles, okay, toward the outside of the cell, and those chromosomes have now separated. All right. So that's it in anaphase. So our chromosomes separate in the middle of the cell at the kinetochore. Lastly, telophase and cytokinesis. This is where we are going to have 2 new cells. In telophase, our chromosomes that were pulled, okay, they're going to get in the nucleus, and the nucleus is going to start to reform, so they're all going to be in one part of the cell, and that's during telophase. And then during telophase, we're also going to have this cleavage furrow here, and this is only in animal cells. Plant cells have something different. So cleavage furrow only in animal cells. Picture this. You got a balloon, okay? You tie a string in the middle of the balloon. Don't tie a knot in it though. And then you start to pull the string tight. Instead of the balloon being like this, as you pull the string tighter, it will start to pull and make an indent in the middle of it. That's the cleavage furrow. You guys know if you tie it tight enough, the balloon will pop. For cells, once it's tight enough there, it keeps indenting until the 2 cells separate, and then we have the new cell membrane formed. Okay, so we did all this already. Good there. We talked about the mitotic spindle. Good there. We talked about the kinetochores already. All right. Okay, so in anaphase, we already talked about the sister chromatids separating. And how they do this is the microtubules shorten. What happens here, guys, is there's like a motor protein on the one side and it reels it in. It's exactly like a fishing rod. Okay? And there is anaphase in all the movement that you guys can see of the chromosomes. All right. And then lastly, guys, at the end of everything, we have cytokinesis. And the goal of cytokinesis is simply to, like I said, divide everything up. All right. Now, we talked about the cleavage furrow. Again, that's only in animal cells. Plant cells are a little bit different. They have something called a cell plate. What that cell plate does, the cell plate, is instead of it going like this, like the cleavage furrow, the cell plate is going to be like a butcher's knife. Okay, so it'd be like having a piece of steak or whatever. Get a butcher's knife, boom, right down the middle, and it cuts it perfectly in half. That's how plant cells separate. So it's a little bit different. There isn't an indentation. Why? Because the cell membrane, because the plants have a cell wall instead of just a cell membrane like in animals. So that's the reason for the cell plate instead of the cleavage furrow. Okay, the cell membrane is a little bit more pliable in the animals, and they don't have the cell wall. All right. So there's a picture of the cell plate right in the middle, and then those daughter cells will separate later on. Okay, so we will stop here, guys. We'll come back and do binary fission, and that's going to be for prokaryotic cells. Just review everything for mitosis, all the steps, and make sure you guys know what happens in each step of mitosis. Have a good day.